السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. رب اشرح لي صدري ويسر لي أمري. واحلل عقدة من لساني يفقهوا قولي. السلام عليكم ورحمة الله وبركاته. Once again, أعوذ بالله من الشيطان الرجيم. We are here with night ta'ala. May Allah accept all of your efforts in this life and in the next. Ameen ya Rabbil Alameen. As we bidnillahi ta'ala conclude the BIOA02 section of our golden Rabb. أعوذ بالله من الشيطان الرجيم. ورب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. My brothers and sisters, inshallah ta'ala, I'm excited to say, but also, subhanallah, um, anxious, subhanallah, to think that, subhanallah, we are near the end of, the final week for AO2 and C17. How's everything?
Good.
It's good.
Yeah. So can I use your book money for a second?
I may need it a bit.
You need it right now?
Not for, oh, for a second?
Yeah, just I need just to write something on the
Okay. Yeah, I know they have even extras there too.
Really?
Yeah.
Let me check
I will go. I will go.
More than mine. Thank you, man.
Subhanallah. أعوذ بالله من الشيطان الرجيم. Inshallah ta'ala, we are continuing with the last six of the Shaitan. Bidnillahi ta'ala, students are gonna, the last class 2, will be inshallah ta'ala looking through the Lemna bioassay that they did or prepped 2 weeks ago. So inshallah ta'ala we'll be able to compare results. SubhanAllah, just like the winter wheat, even though I wasn't able to show it, they do grow, SubhanAllah, and they grow at different ranges, and the goal in this lab, rather than looking at shoot length or biomass of the shoot itself, in this lab they're actually going to count the number of thalli. These are essentially small miniature leaves. They look like buttons or buds on a lemna on this house weed. So, or essentially this type of plant that, subhanAllah, it has like people will be able to, inshallah, ta'ala, look at the effects quantitatively of road salt on these aquatic plants. Because this is something that, subhanAllah, has some, not just ecological, but some biochemical and, safety, or even just, like some important natural implications or effects. Allah knows indeed best. Thereafter, they'll do the mark-recapture experiment, looking at beans and trying to estimate of a sample that they capture and then recapture that how many was available at the original, like how many are there within their tube or task of interest. So inshallah ta'ala, they'll be doing 3 miniature rounds of this to see which of the 3 experiments of the number of marked, recaptured, and captured beans essentially are experimentally changed. So inshallah ta'ala, we'll be doing some recordings, determine the confidence intervals, deduce the estimate the population size, and then relate it to what they indeed would have observed as their true population size within the ta'ala. And subhanAllah, that would, yeah, these are duck reeds, by the way. Very beautiful. I just want to show you. I wasn't really able to show much in the labs together, but inshallah ta'ala, I hope through the visualization, integration, understanding the implications of our labs through AO2 and C17, and inshallah ta'ala get a feel as to what is going on and what could be next to explore for you and those that inshallah ta'ala benefit from you as you learn from these different experiments with us together. May Allah subhanahu wa ta'ala accept all of our efforts. Ameen. And as well for those that inshallah ta'ala are attending and perhaps are actually at EO2, inshallah ta'ala, I also hope these seminars are a benefit, and also inshallah ta'ala that you're able to bi'idhnillahi ta'ala also do the the review, the seminar review that's coming up tomorrow, inshallah ta'ala, April 3rd. And inshallah ta'ala, that will be actually as an update 2:45 to 4:45 p.m. So just to keep a note on that, inshallah. Do want to make sure that we're able to account for salat. So, or inshallah ta'ala, that we, bi'idhnillahi ta'ala, are able to go over lectures or the remaining portions of the review. And inshallah ta'ala, if there's questions, I'd be more than happy to support however Allah subhanahu wa ta'ala wills. May Allah subhanahu wa ta'ala accept all of your efforts once again. And inshallah ta'ala, if you have any questions, feel free to also add them into the comment section. I really, really appreciate, especially for advice, comments, suggestions, compliments, concerns, any outstanding thoughts, extensions. Feel free to, inshallah ta'ala, refer to them. Inshallah ta'ala, I do want it to be a benefit for myself, the community, and most importantly for the Ummah to Muhammad sallallahu alayhi wa sallam, for the pleasure of Allah subhanahu wa ta'ala. So may Allah subhanahu wa ta'ala accept it from you all and allow us to please Him best. For indeed He is the Lord of the East and the West, the Lord of our every test, and in His remembrance to our hearts, souls, and minds, to our hearts find rest. Ameen ya Rabbal Alameen. So may Allah accept. Inshallah, ta'ala, we'll be getting started. Bismillahirrahmanirrahim.
Oh, actually, I think I saw that 80%. Yeah. Yeah. I think fine, you guys can be in the lab, please. Don't go in. One of your
Also, have anyone noticed this is my previous inbox message on purchase? Has everyone got it on purchase? Great. So if any of you, for example, are wondering how like the lab reviews, right? Because as you imagine, you have the lab exam coming up this week, Friday, at 4 p.m. You have 1 hour for it, but my goal is to provide a mini review for you guys. So tomorrow, inshallah, from 2:45 to 4:45, the update will be going over the last 3 sets of God, including this week. Now you guys will be typed up. If you have difficulty either getting through or coming up with you, and let me know. I can send you a code. Which is so small. By the way, is there anyone I didn't mark for attendance? I know Miriam came in. Is there anyone else? Ranran? Okay. Okay, let me step in. Okay. Is Kiomar's here? Vaishnan, Faizan, or Joshua? No? If anyone comes in and maybe I'm unable to see them, please let me know. But once you're done, inshallah, and take your time for the lab quiz. But once the 10 minutes are done for you, you can go into the survey and just give your feedback as to how you felt the lab was with me, what you think were rooms for improvement, what you think was complimentary, or anything really. But feel free to do this whenever you can. The sooner the better. But this isn't timed, so don't worry about having to rush this. And you can also find it on the Quirkus homepage, I believe, in the module section. Briefly, though, this is the outline for today. Essentially, at 10:20, I hope that inshallah we'll be done both the lab survey and the pre-lab quiz. Then up to 10:30, I'll try to go through the intro for today. And then we'll have a bit of time to do two major components of today's lab. So it'll be part two, which is technically exercise 4. And we'll try to do that for about 45 minutes. We may have to, and we'll upload the data here, but you may have to orient yourselves into different groups. Reason being because we're going to be counting the Lemna thalli, so it's going to take a bit of time, but I do want to make sure that all of us are able to complete this in the next 45 minutes. So that inshallah will be 11:15. And then the remaining portion will be counting beans, and that will be done up to 12:30 to maybe even 12:15. So just see if you can budget your time accordingly, folks. I do want to make sure that you have ample time to clean up as well. And if there's any outstanding questions and any components that you want for review, then we can discuss them accordingly. Any questions? No questions? On a separate note, please bear in mind that as this is the last quiz, make sure that you do it independently. As you go for time, you can still leave it open, like your lab manual. Let's try to understand the concepts and see if you need, for example, a calculator, then they're also available here at the middle bench top. Any questions? Regarding lab quiz, today's lab survey. Once again, if you haven't done so, feel free, once you're done the lab quiz, scan the QR code.
Lab website.
That one is
I think you actually minus one is no plus. Because it's minus 3.
oh, I have no idea about that. I'm looking at that.
Yeah. So minus 2 is because it's late, right? That makes sense.
Yeah.
But now it's minus 3. And if
Hmm.
you could look,
Yeah, I appreciate it. Um, right. Yeah, I appreciate I didn't even know that. So normally I should have had that way it goes up. It seems like it went down further. So thanks for letting me know. Can you also, because I don't want to make a so for now, let me before I have a phone. Okay, so if for whatever reason you don't have time, also email me and then I'll tell you that. Okay, thank you. Thank you so much. I hope that didn't happen to anyone else. Folks, I just want to ask if so this is a brief update, and also Joshua, feel free to make your way to the lab quiz whenever you're ready. Um, was there anyone else behind you, by the way, or no? No? Okay. 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 So a few things. Let me just mark attendance. Check if anyone else. Oh, sorry. I don't know if you can scan the QR code. The computer is also glitching a bit, so if it can, don't worry. I'll try to give you a bit more time. I just want to make sure. Then, I'm starting to do a review tomorrow from 2:45 to 4:45, inshallah. So if you can come in, that'd be amazing. It'll be like a lab review for weeks 4 to 6. And if any of you wanted to catch up with the review from weeks 1 to 3, just let me know. But I think we should get started in just a moment. Um, Okay, so it looks like everyone is here actually. Great job. That looks like perfect attendance. So great job, you guys. Now, well, okay. Let me go to Edge. Okay. Or like to our slide. There was one thing I wanted to share. Yes, so I've upgraded or updated the grades for labs 1 and 3. I hope all of you got at least a one scale or one like mark bump up, like a boost, for those three assignments because I wanted to make sure that I'm leveling the playing field so that way I'm not being too hard. And that way, inshallah, you guys get a extra mark. Apparently, there may have been some blip that it looks like maybe some of you, if you submitted late, may have gotten another penalization penalty. So if that happened to you, please let me know so I can adjust accordingly. Because normally I try to correct and like account for that, but it seems like it may have not occurred in the correct manner. So let me know, inshallah, anytime. Sooner the better, of course. Likely before tomorrow would be really appreciated. Um, as well for FLRs, I normally follow the rubric, but I will likely also, we'll see how things go in terms of modulating other um, exercises. But apart from labs 1 to 3, are there any questions for what's in store today? No questions, no questions. Just make space so I can see you guys a bit more. Okay, so let's move straight ahead. Everyone, by the way, able to take a photo, lab survey quiz, or a lab feedback survey. Essentially, really would benefit from this in the sense that if you guys have any concerns, compliments, anything that you want me to consider, then that'd be amazing to know of. And you can scan this now, or you can also, like on your phones or tablets or any other devices, you can also find it on Quarkus as well. So let's go straight ahead. I'm going to go to the lab intro. And so pretty much highlighting the two major exercises today, mark recapture and on road salt. Road salt is something that we're going to look back into the past, right, from last or 2 weeks ago, in week 5. And in this regard, we're now seeing, just like in winter wheat, the growth of these lemnas. But instead of looking at shoot biomass or shoot length, folks, let's pay attention. Instead of looking at those two parameters, you're actually going to be counting the lemna number of thalli leaves within these lemnas. I'm just going to look back and then also show you guys here what I mean. So here you can see there's 3 miniature leaves on the lemnas, right? And so when you're counting, which what you're going to do today is actually look at your cups, count the number of lemnas, put them into plates, and I'll describe that in soon time. But essentially, you're going to be counting the leaves, not just the individual. So bear that in mind. Um, so that way you're able to correct and control for, the number of thalines that we initially started off with. And that'll be an indicator for population growth. Now, when we talk about road salt, right, often it's deposited over the winter season, right, like where we were just the past 2 months ago. But essentially, with that in mind, a few months ago, we are able to, appreciate the amount of road salt that was on, the highways, sidewalks, etc. But now it's really looking at the impact, the biological impact that these essentially deposits have, chemicals or nutrients could potentially have on different organisms, like aquatic plants known as the duckweed or lemna subspecies. And so something interesting to note, subspecies we saw for Daphnia, but also from the Toronto Zoo, we were able to see the impacts on, or just even being able to see different types of organisms. And I briefly touched upon the coral reef, the Great Barrier Reef, and the impact that different environmental deposits could have, pollutants on the survival, not just the reef, but also of many familia of aquatic organisms like crustaceans, the pot-bellied seahorses, the jellies, and many other fishes from the Lake Malawi and other places beyond. So really, that's where you want to be mindful of, like, as these reports indicate, the effects of road salt on fresh water, oceanic water, and other places like seawater, and essentially thereafter on many other organisms that essentially rely or even live in those regions or in those ecosystems indeed. So I know a lot of this is ecologically related or related to ecology in our labs together, but nonetheless, we live in an environment that we should come to appreciate the different organisms around us and to see the impact that we have to share, hopefully for their betterment and not for their detriment or detriment. So bear that in mind, folks, as future biologists, scholars, polymaths, in your fields of interest or specialties. So let's move forward. Exercise 3 will essentially be done post-lab. So you can actually access this on Quirkus whenever you're ready to prep up for your last assignment. And that's where you'll essentially make use of this in referring to details that are actually mentioned on pages 89 to 90. So if you want to have a look at the lab manual, you can look at it now because that's where we're actually going to go to the next week exercise or the next upcoming exercise, which will take us to exercise 4. Before I move forward, any questions? No questions? I'm sorry if my timer looks a bit off. I think with the charger, sometimes it gets a bit techy, but nonetheless, exercise 4, the goal will be actually taking out your lemnas. So each group, let's see, how are our groups now? I'll read the instructions, then I'll let you know. Pretty much, we have the same, containers from before, the 3 reps for the different conditions of the control, the road salt concentrations, 5 of which we explored as separate groups in our last lab. And so the goal of this lab is really trying to count out the number of, I thought there was an extra slide. Maybe there isn't. Okay. So pretty much your goal is to essentially count them out onto those 3 different plates. 1 plate in each of your blocks will be served for 1 type of, for 1 rep essentially. And my advice, both, let's say, I'll grab 1 as a demonstration. You guys can all come to, actually stay where you're seated. I'll come to these spots and we'll make it easier that way. I'll help Aaron just for now, but I hope that yeah, these are okay. Different which okay, but okay, I want to think about this a bit more. Essentially, some of the results are different from other labs. But I mean, I'm referring to last week. Last week, we had a lot of numbers. So you guys look like you had it easier, which is good. Or like hopefully applicable. Let me just see what's our next step. So you were in control, right? Who was control? You guys were control? Yeah. Okay. Okay, so how about this? I'll like for each group have 1 member or 2 members provide like their cups, and then I'll demonstrate what we need to do next. So I'll have 2 members per group. It may technically be the 2 of you. We're gonna go to exercise 4 first. So just bear in mind, once you're done, that'll take about 45-ish minutes. If you guys have questions, let me know. Make sure you grab your benches or your blocks concentrations. And that will make it easier. It's okay if technically another group is using another set.
And we'll talk about the
No, that's 19. Yeah, that's why I did. Yeah. I'm positive because there's the piece of tape too, and there's the one lady told me if you don't need tape, you grab the cup. I'm positive this is.
So let me just give you guys some advice. You guys are going to be using essentially these yellow inoculating units. You have to use them in microbiology, which are also needed. So you think about the units of, bearing in mind, contaminating the surface. The idea is that having, for a moment, open up one of your tubes, or sorry, one of your so microbiology tubes. Yeah. In this class, we're using cups. So I want you to open one of them. And the goal of my advice to you guys is to take one out at a time, but accumulate the number of thalli that you see. The thalli, once again, are miniature leaves, shown on the board 7 for 2 lemnas. And my advice is to keep counting up. So take your time, but try not to separate them like in your count. So don't do like 3 and 3 and 3 and 3 and 3, but rather essentially count up. Let's say I'm counting a got a lemma and counted 5. I'm going to put it in one location, one edge of the plate, right? So that way I can keep things organized and they keep going up or move around with the plate. But then I'm going to keep counting. So I'm going to go 5, then you see another number that has 2 leaves. That's going to be 7. So 5 because 5 is 7, right? So then I start off with 5, I put 1 down on one edge of the plate. So that way you can separate them accordingly. Then I count 2, so then I count say 7. And I count another one that has 3 leaves, and that's 10. Then one has 4, 14. Then 6, 20, etc. So you want to keep counting them that way, so that way you don't have to take them all out on your plate and then recount them. You can count them with efficiency, keep counting up, and then if you worry that, oh, let's say I need to go back, think about what last number you said. If it was 3, then accordingly, either you're right or you may adjust your final number. But just try to accumulate. So go up in number as you count the number of thalli from each lemma that you place onto your plate accordingly. That's my recommendation. If your group is done, we'll place them onto Excel. But I do want to mention that if your group is also done, please let me know, not just the number of lemnas that you have right now, but the number of lemnas that you may have had from the past 2 weeks. Because some of you, I mentioned that, okay, you have X number of leaves, maybe there were 12, and maybe this will be a good time for you guys to see. From my previous week, or actually, let me go to here for our lab. We typically have 12, right? Because if you recall from 2 weeks ago, you would have had 4 lemnas, each with 3 leaves, right? So 4 times 3 is 12, right? So 12 for you guys. Important reminder though is that if, for an example, you had, let's say, 4 thalli or 4 leaves on a single lemna, and if that number did not like add up to 12, but maybe to 13 or 14, then let me know so we can adjust it from the initial count. And that way, essentially, the goal is that you, inshallah, will see for each treatment the different categories of the number of thalli, and we'll input them as a group, and then I'll talk about stats on what you need to do next. Any questions? No? So let's get started. Let's spend the next, 45 minutes, I'll say up to 11:15, to go through the rest of the practical. For this exercise. Once again, if your group is done counting, please help other groups because some of them may have a lot more in number. Some of you may be done in the next 10 minutes, but there's some that may actually be done after 40 minutes. So try to help them out. And if anyone wants to swap now for the number of lemnas that they have, let me know. Otherwise, we can also stay where we are. And then when groups are done counting, they can let me know. I'll input it into Excel, inshallah, and then you can help out our other group members. How does that sound? Sounds good? Sounds good? So on your marks, get set, go. And feel free to let me know if you have any questions. Yes. Folks, make sure you count them for each replicate. So each replicate will be done separately. You can write them down in your notebook, on your phone. Just let me know, and then we can count them accordingly. And input what you put. Also remember, it's the number of thalli. So let's say this is your plant, your lemna. The duckweed has 4 thalli, right? So just don't say that you have like 20 different plants, but if it has, let's say, 80 leaves, then that's what you're counting up. If you find it easier, there's also the counters. So you can actually take all of them out into your plate and then count them with the counter accordingly. If you find that easier, that also works. But let me know if you have. I know some people are also counting them directly in the cup. That is okay, but I would recommend to be mindful that you may not be able to see all the leaves. So try to actually count them outside of the cup, confirm at the very end. So let's say you're counting the cup itself, and think then if you have a number, let's say 56, take them all out. Thereafter, onto the plate separately and see if you get the same number. Just so that way you validate them. Without running the risk of So, folks, if anyone needs me, I'll be down at this end. Something else, counting some numbers with you guys. I'll just fit it too. Also, if you find small bugs, you can include them into your count to make things consistent as a group. If also you guys need a timer, I recommend trying to see if it can be done in the next 30 to 40 minutes, 30 to 30 minutes ideally.
Yeah, it's only a difference in 15.
Yep. If anyone would like to swap with me as I go into itself, I would appreciate it. So you are done? You done? One more. Okay, so I'm gonna ask something, something, one. If you want to choose the on count of your values, the one I was counting, I was talking that's a hundred and two thousand. So one or two, that's a hundred and two. So I stopped. It's very little. Yep. Yes, the numbers are all covered. Firstly, oh, there is one time, we are actually using, so we can, if you want to do, you can go out, make note of the, then you can,
Yeah, I don't know.
you know, appreciate it. So yeah, that's the one. 102 is where I am. Um, yeah, so for context, they're the control, right? So they wouldn't have any root salt added, which is why that they grew up naturally to have an abundant amount of sunlight over the past 14 days. So you want to think about, like, as we're going to compare the results. Yes, you guys can, and that's actually the benefit. I was thinking that one. So this is starting at one. You guys can talk out individually and then see what does that. Any questions? You can figure it out. Hey, Charlotte, what group are you in? Or what was your results for your group?
We're doing
1.25. Can I get your values?
A was 236,
236,
B was 252,
252,
and then C was 84.
84, really low. You think so?
Yeah. I didn't look really cold. That's why I actually counted. There was a disappointment.
Oh, okay. But the other ones, were they counted on in the cup itself, or?
Yeah. On the tree.
Yeah. No, on the plate. So on the
Yeah.
plate. Yeah. Okay, that's good then. That's good. I just was worried. Uh, Perfect. So now let's go to this one. Wait. Are there any other groups that are done? How's it going for your groups? I would say like these two groups. How's it going? You done? Yeah. Okay. What concentration of road salt did you guys have? What was yours? 5, 10? Okay. Can I get the values when you're ready? 75? That's for rep A, your first one. C, okay. And what's next? Or B or A? 66 for B or A? B, and then 76. Okay. Okay. And now I'm just gonna find out from where these folks are. Okay. Um, oh, sorry. 6. What are your other results? No. Is this a 0.625? Which one's your concentration? 5 grams? 5 grams per liter? Yeah, okay. Um, don't worry, you'll see the values. It's going to be posted on Quirkus. Okay. Actually, one moment. Yeah. Maybe I should get Aaron's first. Kind of 0.625. And what values were 123? 158. Okay. 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 Um, are you sure, by the way, in terms of values? Yeah. You know, I ask. Well, I mean, it is possible. Let me see. Let me have a look. Again, if you don't mind, for reps A and B. 73, 221, 166. Okay. So the trends aren't looking like, generally speaking, they are moving down, but it's moving a bit up as well, which is why I'm a bit concerned or like wondering. I'm not really too concerned, but it's something to keep in mind at least. If we compare with my previous lab, you'll see that the values are actually quite similar to a degree for 5 grams because it seems like it goes up, which is interesting. If you can compare here, you notice that it goes a bit up at 200. Even though the values are not the same, the trend is quite similar and it may be corrected for in the control. So we'll see how those values relate. But you're only tasked to focus on this table and essentially completing the rest of the results for the growth rate, mean growth rate, for all three reps, and then the percentage growth. For Charlotte, do you have Aaron and Charlotte? Do you guys have the last rep counted? What 123? The exact number? Are you sure? And that's the third one. Are you guys taking out more than one part at a time? Or were you guys talking individually? One by one at a time, right? Yeah. How will you know? Yeah. The reason I asked this, I'm noticing the complicate one together. I'm noticing more than one here, 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 here. I'm worried that maybe we need to especially compare it. So it's already done. You can speak out these are the parameters. Separate more. So it could be like maybe
like I was like myself before.
Oh, the info.
Yeah, I did.
Oh,
Yeah.
this is it. Yes, right. You're like, give me the wrong place. My one. Then it should be your looks like right? For the whole, like, over on the like in exceed for separate.
Yeah.
That was her one, but that is mine, but she can't.
Yeah.
So, let's count them this one for everyone. No, then I'll give and then she can go on yours. Don't worry, you know, to be honest, you have to do the easier stuff. That's usually the one. I don't know. One day. So how's it going? And that's the control for 605. Okay. Did you why do we did you include the 102? And that is B or C? I don't know. B, okay. 720. Okay, any other groups that are missing their values? I think we're waiting for 3 more people just to confirm. I'm happy you guys are actually ahead of the game. Keep it up. And we can maybe be done in the next 10-ish minutes with this exercise. And the last one will be, I hope you guys will enjoy it. Will be a bit. When you guys are done, actually, there's 2 things to bear in mind. This is your last journey. For this one, when you guys are done,
officially done. We have a few things. One is canister to actually deposit the lemnas. So essentially, use your plate and please use the water on water, pour down to drain the lemnas down into this bowl. And then thereafter, the lemnas should stay in this location. Don't put them down the drain. Please put them into this basin, and then you'll take your cup. Essentially, you'll stack them all up here, and then the lids go here. They should all be unlabeled, so if you have the marks on them, just use ethanol or like a French Puree to remove the excess marks, so that way we use. And then we'll place them all down side together. Just bear that in mind, folks, as well, that you should have lemnas, so that way it doesn't obstruct other people's observation. Just bear that in mind. Any questions as well? If you are, by the way, like, if not, just let me know, like, if you're free, in case of it. But for now, let's get back to the. In the meantime, have a read through our last one of our major exercises, exercise 2, which is going to be the mark-recapture, which is going to take a bit of time to talk about. So I recommend looking into the final exercise, and I'll give you the page numbers for reference shortly.
Or if there's any questions, also let me know. This is part of Lab 6A, and in reference, these are pages 80 to 82 that you'll be focusing on. So have a look at those pages. That will be our next exercise. How's it going, madam?
Good. Clean.
Great job. So once your group is done, please make sure that you help the directions that you know. If and trying to know that you use and then they're actually using the water.
Yeah.
And we ask them. You can slowly pour the water so that way all of it goes down. And if you need excess, then you can use a little bit of extra water. But that way we're saving water, we're not wasting it, and that's also very beneficial.
Yeah.
Oh, can I get an update? Aaron and Charlotte? Yes, when you're done. When are you done? So, oh, did you count them?
Yeah.
From there though, or
Yeah.
are you 100% sure they're right?
Yeah.
The issue is sometimes the lemon is sticking up. Okay, 200%. We'll keep it. What you're gonna do is pour them down into the bowl there. Yeah.
So is it also to record it?
Yes. No problem. It's yes, that'll be not a problem. Email me though, and I'll try to see. No problem. They need the lid. The lab techs need the lid. So, yes.
I recast it for
I'll help see
yourself. Yes.
with ethanol. Did you clean it with ethanol? Yeah. Okay.
That when I do, I like look at each individual.
That's an 84.
Yes, yes.
Okay. So that's 1.25.
Hmm?
Which concentration was that?
Oh, 1.25, yes.
And then 119.
Yeah.
Good job. See, 117. Okay. Okay. For the last one for 0.625. Okay. Perfect. Perfect, you guys. So, just this is a separate point, but we have variation at times. And this is why I recommend just to make sure that you guys are, following with addition of, essentially placing in the lemnas onto your plates and individually counting them per lemna. So that way you don't run the risk of overcounting or undercounting. That way our values will be quite reliable.
No, because that's I'm looking for why they're just using.
So that's a good question. I think they technically find the lids more reusable, whereas the cups themselves, students always have to label them. So that makes sense. And then pretty much the lids themselves have to there, and then the cans always have to afterwards.
So is this good enough? Because
Yeah, that's good. That's okay. Was that the 128 one? Great job. Keep up the great work. What? No, it's okay. It's alright. You're an amazing colleague in the Bible. Yes. 592?
Yeah.
Perfect. So essentially, I think I should have said that actually. Not to go on about. I hope you guys become somewhat for longer. You guys will become like the next gen of all. Maybe next week, because it's also exams. But nonetheless, don't worry, you guys keep talking. So everyone, are we all done? Are our plates also dry, by the way? Can we dry up our plates? Use the paper towels, we'll dry them up, and then return them back. You can use the same paper towels together. I see a bunch that are there. You could use those same paper towels. And then in the next two minutes, I'll talk about data analysis. There are paper towels also there. Once your group is done, for exercise 4, let me know, and then we can begin shortly. Are our plates clean? Our plates are clean? Yeah? Yeah? All plates are clean. The cups are put back. Yeah? Please make sure as well that the lids are placed appropriately as well. Any questions? No questions? No questions? So let's go forward. So as you can see on the board or on the slide, on the screen, at least, you can see that for each treatment, from the control all the way to 10 grams of 10 grams per liter of NaCl, sodium chloride, what you can see is, inshallah, not just the base or initial number of thalli, which is going to be very important, but also and this is something to keep in mind, the values or the number of thalli or leaves that you found for today. And I want you to keep in mind the units. I also want you to keep in mind the trend of the numbers of thalli that you see across treatments. So around here, you're generally seeing around 600 to 700. And then it goes down from the control from around 6-ish hundred or 650 roughly as a mean, all the way down to like 100 to about 200 to then 100, then around 180 to then thereafter below 100. So you could see that the trend is going a bit up and down. And this is interesting because the same trend is actually similar to what I've seen. But what we're really going to focus on, is rather the data analysis. And I'm just showing this sample from last week as an example of what you would do. So for your data analysis, what you would actually be doing is essentially following through this formula. Essentially, you're doing this in ln. And so the ln is, like the formula goes as this. And I'll show it on the next slide on your page for the practical, which you'll see also on Quirkus. But pretty much I'm following up with the same formula. This is the natural logarithm, so bear in mind you're using ln. And then you input the first, or technically the last number of thalli on day 14, which is N2. And then you input that, close the bracket, minus ln again. And then you are subtracting it by the initial number of thalli. And you're closing the bracket once again. And then because technically there's a bracket here, you're going to close it once more, the entire equation. But then you're going to divide that by the time. And the time is 14 because there's 14 days between N2 and N1. So you're dividing that by 14. And that's what you get for your growth rate comparing N1 to N2 across 14 days for your first rep. If you want to see those results, you can repeat the same equation, or what you can also do is essentially paste down the same equation across the reps, and that's what you get for your growth rate across all three replicates. Thereafter, and by the way, does that make sense, everyone? Everyone paying attention, by the way? You can see some of you on the screens, but I want to make sure you guys are able to see this as well. So for then mean growth rate, you're essentially taking the average. And I'm going to keep this for you guys for like, I ideally take note of what I'm saying, but you take the average essentially of the mean growth rate that is shown here across the three reps. You press enter and the ending bracket is included. So then that's essentially your mean growth rate. Now for the control, actually, let me repeat the same process so you guys can also see this. Not just for the control, but also for the next experiment. So this is essentially, just so you guys can understand this, the same for this set of treatments. The growth rate is essentially translated. So if I went to here, it would be the ln of N2 for this rep in 0.625 grams per liter of NaCl. Of sodium chloride, NaCl, and then you have the initial base in ln, and then you're dividing that by 14. So the formula continues as you go all the way down. And then what you want to do is repeat that process, average. If you do this, by the way, I don't want you to do like this same setup. Actually, it works here. This is perfect. Sometimes in Excel, it will actually take the average of the following three cells. So it wouldn't give you the full corrected average. But nonetheless, when you're comparing thereafter the control is assumed to be 100%, right? So I'm putting 100 here. technically it will be 1 because you want to make sure the units here are in percentage. So I'm updating the column to make sure it shows you as 100%. 1 is the decimal form, right? So 1 out of 1 is 100%. And then when you want to find the control or the relative growth rate in percentage, you're putting an equal sign divided by the treatment that you're looking at, relative or by the control treatment. So bear that in mind when you're finding the control growth rates or the relative growth rates. I'm going to make that correction right now. Actually, I'll save that afterwards. The idea is that for your treatments, so you would have calculated, right, from the formula here, the growth rate, but then you would take the mean, as we did from before, and then thereafter for percentages, always put the equal of, like, equate or essentially take the mean growth rate of your treatment and divide that by essentially the control, always. Don't do it by the other treatments. Always divide your experimental by the control itself, which would be here, the control's mean growth rate. And that's what you get. And then essentially you would get values accordingly. So I hope that helps. If there's questions, let me know. But once again, you're essentially doing this for all of the other cells. And then you're going to be showing this data as well as a graph with a detailed figure caption, with the axes, title, units included, inshallah, for your lab 4 assignment. Your lab 6 assignment, but from exercise 4. Are there any questions, by the way, doing this data analysis? Does it make sense? Yeah? Okay. So just bear these values in mind. Note the trend as well, and that's going to be very beneficial for your understanding of the rest of the practical. Also, even though like the values are a bit different here, you'll notice the lawn, generally speaking, has similar growth rates at the very end. And that's actually the unit that I wanted to put for you guys. So now you can understand that it's essentially percentage growth rate relative to the control. And that may make it easier to understand for you guys. So with that, once again, any final questions? No questions? You will be submitting this for your final assignment. So make sure everything is, make sure you understand the material. If we're ready, we're on time. Not now. I'm gonna go forward and skip actually. We're a bit behind by a minute or two. We'll go to the population dynamics, and really populations we are already aware of, but essentially they are impacted by stats of or the numbers of births, deaths, immigration, and emigration from and through a population. So in and fro, or to and fro a population. So you have birth and immigration, surplusing or increasing the population size, and then emigration, i.e., people are immigrating with hijrah, like going away from a specific region and then going essentially the total population size with mortality can decrease. And ecologists are interested in this. And as we talked about at the zoo, integrating these concepts once again, conservation efforts are in place for certain organisms that may be reach towards extinction. So these are things that you want to bear in mind, right? Some populations go exponentially upwards, but some also decline, right? We saw like the Solomon Island frog or the skink or the black mother, or sorry, I remember we talked about the Omer tiger, right? So you can see that there are different, even like here on the slide, the American bison, there are different conservation efforts in place to make sure that previously endangered species could thereafter thrive in the right conditions, which also connects us to thermoregulation, because you want to have these organisms in the right habitats, following the right within the right pretty much thermostat in the right thermal regions. So you want to bear those details in mind. And even though it's a bit separate, but these different populations have greater impacts on communities, which make up those populations up across diverse organisms. So like the Toronto Zoo, for example, is filled with populations of different organisms and makes up an ecological community in a single pavilion, and then an entire ecosystem in the zoo. So think about those details. You go from the ecosystem of the zoo to a pavilion as a community, and then you have populations like the Solomon Island frogs, maybe, or like the jellies because they're very abundant. So you have a population, which for definition here is an individual of a given species, all individuals of a given species that live and reproduce in a particular place. So think about like those specific habitats, think about those specific, niches, or like closed, enclosed areas. Those are like populations, and then going upwards to communities in a pavilion, and then into an ecosystem like the zoo. So, and even outside of the lab, right? Here at UTSC, you go to the valley, you go even near Sam Ibrahim. If you go to the garden UFT has, you'll see many different types of populations, and perhaps if you scale up the entire ecosystem at UTSC and perhaps at our campus network. So think about those connections. Now we're gonna go, down to the very bottom. So we're going like down under, and we're gonna really be dealing with the mark-recapture method. And does anyone know what that method really highlights? Does anyone know a bit about it? Based on what they see, exercise 2, pages 80 to 82 of the lab manual. To go back, pretty much it's an estimation. So think about like this. How do, biologists know, or like how would you be able to determine like the population, let's say of bison or birds, or in our case, beans, in a specific, population? Our goal is to essentially follow through with mark recapture. And the idea is that we're going to be doing 3 sets of mini experiments or methods. And essentially, we're going to be trying to capture beans, right? And I'll talk about that soon, all in one bench. We'll probably do it in the middle. And essentially, the goal is that you will be essentially following through with what you see on page 82. There are specific details on the next slide and on page 81, sorry, 80 to 81. But I just want you to understand that primarily you'll focus on 3 different parameters: M, C, and R. The number of marked beans, which in your case will be 20 for our first 2 experiments. And then the number of captured, which is, or sorry, in this case, total recaptured or captured, both are sound for the first experiment, which is essentially the number of beans that you're going to capture with cups that you have on your benches. And then the number of, and the last parameter, R, is the number of marked beans, which you would have labeled for 20 of the beans in your vial. And the number of marked beans that you recaptured. So that's something that you want to bear in mind, that essentially you're going to be capturing a number of beans. Some may be marked, some may be unmarked. And so your goal is to make note of all 3 parameters, which we'll be exploring in tandem today. Any questions before I move further? Okay, so let's get everyone ready. I'm gonna everyone can come by. So let's turn to the middle bench, and I'm going to describe what you guys do. Basically, ahead of the class, and keep up the work. Your goal is yeah. So you want to do 2 things. One is count the number of beans. So this is a little bit. You want to count the number of beans and count all of them. That's going to be your last number. That's row piece. You can count them here now or at the very end. But the idea is that you count them up
put them into page 18, the lowercase n that you find at the end of the table. And then you're going to label 20 beans. Try to label them so you use the marker that you have in the Sharpie. Try to label them across the country so that for whatever reason, and you can do it more than how I'm doing right now, I'm just being a bit conservative. But essentially, you want to make sure that if you were to look at either side, you know this is the mark bean, right? So that way, that if I drop it, I know it's mine. Right? So you bear that in mind. Do that for 20 of those beans in your or sets of 2. So you're going to do that. Once you're done all of that, or once you're done labeling 20 of them, that's going to be your first M1 for that table there. So you're going to have 20 roughly. And then when you're done, you're essentially, okay, let me make it easier. I'm going to label. Easy means because I want you guys to see and know what we need to do. So this is just an example. This is for Ramadan because also she is going to be doing by herself and going to make Joshua. And likely for Aaron as well. So let's see. Hopefully you guys can be able to do this. But I have 3 of them labeled, actually 4, 3, 4. And then the goal is essentially, when you're done, you place them all here, you close it up, make sure that it's fully closed, start the space. Give it a good space, open it up, and the reason I but the idea is that in the middle, if your cup is broken, so let's say you had all X number of beans, you much n in our case, or n. All of your beans would be scattered about randomly, and ideally in the center. Put your cup in. That should give you both rice and beans, both marked and unmarked. And then you're going to slide this from plexiglass or this plastic slider essentially at the very bottom. So that way it goes underneath your cup. You may have to lift it up just a bit, but eventually you turn it a bit on the side. And then, because technically this plate would have been empty, you place it into here or into your white box, so your white container, and then essentially you'd open it up and see the number of beans you have. You would count them out, make sure they look all around. And in this case, this is unmarked. Let's say I had more beans of marked and unmarked. That's where you're going to include them for C and R thereafter. So the total number that you would capture, which eventually you're going to repeat this until 20. So that's where C1, which is shown at the top, would equal to 20 roughly. So you're going to repeat that process. Do this until, like, you may only count like 3 or 4 in the first cup, but you're going to repeat this multiple times and keep taking them out. Don't return them back, but keep taking them out from the ones that you captured. And in total, count them until they reach 20. And then if they reach 21 or 19 or 18, that's okay. Keep counting until they reach roughly 20. And then for R will be the number of marked beans that you reach. Does that make sense? That will pretty much be actually experiment 1. The other sets of experiments are relatively simple. They're just variations of this one. And I just want you to be able to do this once again. And when you're done, let me know. But once again, you're taking out all beans, count them up, mark 20 of them, place them all in, put cups and count or take out, capture at least 20 beans. And then for R, you want to find out, you want to count the number of marks. And then you'll write that down on page 8. Any questions? Questions? On your card? Okay.
Yeah.
Return back the rice oh, it doesn't add a confounding variable, and then you could redo the steps, clicking up again, and find that you recapture the capture. Make sure, by the way, labeling both sides to the 20. If anyone is missing cups or cups that they notice that are broken, please let me know. And you guys can also work as if that helps.
No, that means from this.
Yeah. Aaron, how many beans have you labeled? 7? Okay. So, Aaron, you can just stop where you are.
Well, we knew it should be, but ours might be like fun. I mean, we have to stipulate. So what are you at? Up to 5. Good enough. Can you take one of the markers?
No. Yes, so for experiment 1, you're gonna mark 20 beans from the 70 or more that you have. And you want to make sure that once you've done that, you shake them into the container, basin, mix it up, and then take out per cup up to 20. So keep cupping out, but make sure that you put back the rice each time. So that way it serves as if the population is still intact for essentially the other organisms involved. You have a population beans, a population of rice, filling in a community. Everything okay?
Yeah.
What did he say? Well, 69 is okay, but this one, 69 will be at the board case. So, folks, just so you are aware, the lowercase n is the total number, the actual number. The uppercase n is the one based off the formula that you'll be using from Emma, C, and R. So please bring that in.
Okay, one.
It doesn't matter how many stuff you use. You just gotta keep using it until we get 20 beans on that plate. You see 20 beans on that plate. But it's coming. It's gonna be, you can put it here. So you can dump the rice back.
And yes, if you use the plates, if it helps sort out each time you put the cups out, and then use a separate weight bowls to essentially take out any beans. Either way works, but just make sure that you capture or see about 20 beans, which can be seen on the screen.
So you put the rice back in, you do this.
We'll do that when we have on 20.
Yeah. And by the way, separate points, so please make sure that you don't fill the rice on your benches. Make sure that you fully close your lid without putting things that are
You play with 20. That's for a different group.
Mark. Second one is we put these out and take out another one. Yeah. We have three. Like how we get pulled out from the coverage.
Yeah, that's it. Okay, so you just step now. With this, you should be essentially putting that as work instead.
Okay, that's
Should be more, it should be like 66.33. That's about. Five, 7, 3, 4, 5, 6, 8. 8 for R.
Are you sure? I don't know.
We got it right.
And then take another one, okay? And then capital number to be smart.
Okay, so these stay out.
This one,
These stay out, and we do the same thing again. Combine it. Yeah, you don't remember. Or the next. Yeah. Right. 24.
Until you reach.
I have it here. Yeah, the next one is actually the yeah, absolutely. It's already 10 more. Right, but it's not. A lot of rights.
So, folks, let me now go to experiment 2. Are you guys already there, or seems like some of you may already be ahead? If you're done, experiment 1, determining M, C, and R, and the lowercase n, which you would have done at the very beginning. The next step is actually looking at page either on the slide or even from the table from before. The goal is in this step, you still have the 20 that are outside, right? Right, likely from C1. So your next step from the first experiment to the second is to still repeat the process. So it shows in page 81, pretty much you're recapturing. So you're still repeating, continue to recapture individuals from the population until you have removed approximately 20 more beings, essentially, right? So you don't have to put them all back for experiment 2. You're just repeating the same process because you still should have some unmarked and marked beans inside the rice container. So your goal is just to repeat the process until you get 20 more or 40 in total, and that will be your C2. Your M2 remains 20, by the way, right? Because you haven't adjusted labeling any of the other beans for experiment 2. So your next step is really continuing C2 to capture 20 more beans
total of roughly 40. Ideally, get 40. And then R2, in this case for experiment 2, will be the number of labeled or marked beans that you've captured in that total of 40. So pretty much we'll be combining these two in this section. So just bear that in mind. Any questions?
Oh, that should be, yeah, that's pretty wrong. We did do 20. That is and 13 unmarked, that's what it was saying. Right? And 8 was 13 to 21.
Yes.
So, yeah.
So you guys had 4, 3, 1.
Yeah.
That's okay. Yeah, so it's okay. Based on a vision, but it's not like initially subtraction.
Yeah.
Because the total number of things that you have to. And then so what that makes sense to me? Essentially, it's no, primarily you're focusing on-
Yeah. Between 2021 8 and 2042 13. That was our thing more. 2042 13.
So the total one.
Yeah.
I'm gonna revolve them.
13.
Combine all the through one and
Yeah. No, that's done. We gotta mark 20 more. And then do it again. They're all come back in, but we're doing the exact same thing, just 20 more.
So, to clarify the question about experiment 2, folks, let's pay attention, please, because I think there may be a misunderstanding. So I'm just going to draw it out so you guys understand. So you have experiment 1, right? We did M1, roughly 20, right? Total marks, right? This remains the same for experiment 1 and 2. Then for R1, which is the number that you captured, so captured in cups, we'll say. We'll say total captured in cups. And that should also be roughly 20. And then C, for each group will be different, right? Because you can have maybe 8, and I'll actually highlight what this refers to. Total marked beans, or number of marked beans. Right? Now, this is a variable value, right? And something I want you to bear in mind is that you would have likely already got the number. It could have been 8, 13, 7, etc. If you have a lower, like if you're within the range of, I would say, 7 to 13, that should be okay. If you're lower, let me know. But if you're a bit higher, that could be okay as well. M2 is also, once again, the same as M1. So it will be roughly 20. Now, R2 is you repeating the step, meaning that you are taking an extra 20 here, right? So bear in mind, the 20 beans from experiment 1 are still left outside, and the rice should be put back in. But now you're repeating the step of taking out beans and capturing 20 more. So you do the math, right? 20 plus 20 should be 40, right? So you should technically have 40 here. And I'm saying this because I'm noticing some of you have 20 on this location, but this should actually be 40 because you can imagine 20 plus 20, right? From both experiments, you would have taken out 20 means each time, gives you about 40, right? Depending on how many you took each time. So you would have likely gotten 40 there. And then your C2 is essentially the total number of beans that you would have gotten from both experiments. So please bear that in mind. This adds up, or you're essentially adding up in this regard the total number of marked beans. So this is, remember, the total for C2. Any questions? Does that make sense? How are we doing so far for experiment 2? Are we almost done? Yeah? If you're not done, let me know. Who's on to exercise 3 or experiment 3? You guys want experiment 3? Experiment 3? Okay, experiment 3. Is your group on experiment 3 or 2? Both sides or? 2? Okay. By the way, did I answer the question that you had? I don't know if I recall it. Yeah. Okay. Let me ask.
Yeah.
Sorry, folks. Brief error or mistakes that I want to clarify. Technically, so I was reading in the wrong order. This is C2. C1 and C2. So just bear that in mind. That's your total for the number of recaptured beans in each cup. And then R is the total number that is marked, which essentially refers to recaptured, right? So recaptured beans. Bear that in mind. These are your values of N, C, and R. And then you should, inshallah, see the values that you would have gotten for each of the different maps, so, or different parameters. Let me know if that makes sense.
Well, it's at the end. I don't know the view of we have 26. I can't wear your grade of my favorite. Yeah. Okay. Yeah, that's fine. $17. How many total? Okay, so yeah, you're gonna mess up with it all.
Yeah, okay, perfect. Now, okay. Do you have restart on the beginning? It's only the one now. How to do it?
Looking back and forth. I didn't see it. I mean, if you pick the strategy, did you look it up? Like, because if you think like that, that's pretty big. There is the 60? That's okay, we're doing 30. No, that's too late, I wrote it down. 40, 39, 23. I never done it. Yeah, it's all hard. All the beats are. So. I don't know. Yeah. Yes, we basically just follow up first, then we'll do the backing, and then take one round and then take one.
Okay, I get it.
So yeah. Well, it's the first.
All of them. All of them. Okay.
Huh.
No.
Yeah. Yeah. Yeah.
Wait, you
That's a free loss of it. True.
Any questions from home? Okay, let me answer a question on experiment 3. Some of you have already gone forward, but let me just briefly say the experiment number 3 involves you essentially now marking 40 beans, 20 of them that you already have labeled. But when you reach the stage, you don't have to rush. We have a bit of time. But essentially, once you're done, noting M, C, and R for experiment 2, which should be for C, 40, R more than your experiment 1, and the total and the number of beans that you got from exercise 2 that are marked, but if a combined total. So a combined total for R2 is something that you want to bear in mind. It'll be something that I'd be able to note. But after that, experiment 3 is essentially labeling 40 of the beans that you now have already outside from experiment 2. When you've done that, place all 40 beans. Oh, this is a separate point. Sorry, this may have actually been miscommunicated. You may actually, instead of having, so I may have said, yeah, you currently have 20 marked individual beans, some of which have already been recaptured and some are still separated. So what you could do instead of actually labeling 40 new sets of beans, just label an extra 20, right? Don't label an extra like 37. Label an extra 20 beans. And that way it'll come up to 40 as your new M. M3 will be 40. And then you're just putting everything back inside, cupping up to 40, and then inshallah you'll get your results M, C, and R. Does that make sense?
Well, I'll look through the more. Can I ask you a question about the calculations? Yeah, okay. Is
Folks, I want you to look for cleanup. This is what you guys are gonna do. Pay attention, folks. This is your set of beans with rice. Into this canister. Take it up until you get all the rice out. Back home for us, actually, you actually do this for settling flour. So you want to get quality flour, make sure that there's no residue or the green inside. This is what you kind of do. For many of you that are maybe from like the Middle East or the Asian continent, you'll probably see many of these as well. But essentially, you'll use this and essentially filter out the rice. And if any beans come out, essentially we put it in. This is for the end goal of experiment 3. So if you're not done, not a problem. Just once you're done, place your rice in here and with your beans. And then the beans you'll eventually place back into the vial. And the remaining amount you'll place into your container here. If it's blank, then you don't have to. Just try to take out all the rice, because sometimes you'll have it come out. And you want to make sure that the bench is not clean. This will be your semi-final with me, so till the very end. So pretty much cleaning up is relatively simple, folks. When you're done, let me know. But I also want to see your results before you conclude.
I don't know.
How do you do it?
Yeah, it makes sense. If the world, if you can answer this fire question now, that's pretty close. That's right. Yeah. Yeah. But also something good, it's like, very fast. That's important. Kind of. No, the focus right here. I just wanted to. Yeah, my experience always like to make part of it very like. I was like, this is if you need to reshape this. But it's
Folks, I'm going to show a formula to make it easier for you to understand. Very useful. Can you just remind me? It is M divided by N. And then R will C. Okay.
Yeah.
So perfect. Folks, I just wanted to get your attention just so that way you're able to understand how you calculate N, uppercase N as your estimate for total population size. Look at N being at the denominator, right, on page 81, I believe, in your lab manual. Because this is what you want to solve for, right? You want to actually isolate it. So technically move up, R would move down, and then C would move up here. So you would have M times C divided by R. Now, for exercise 1 and exercise 2, both M's are roughly 20, right? Each group will have a condition or parameters, but you want to make sure that you're solving accurately. Why? Pay attention as well. That M is normally around 20 for exercise 1 and 2, right? And then C is going to change. For exercise 1, it will be 20 from the number of beans that you've captured in the cups. And then for exercise 2 and 3, it'll actually turn to 40. Right? They have different procedures, but hopefully, if you've reached exercise 4, you're aware that you would have marked your beans up to 40 in total, place them back in. And for experiment 3, you would have essentially cupped out 40 or captured 40 beans using the cups to derive the total number of R. R, which is what you're going to solve. Sorry, R is the number of recaptured beans, and that should be for experiment 1 and 2, if I'm not mistaken. Oh, that's actually variable, as I mentioned. So R is something that you guys will see based on the number of total marked recaptured beans. So you want to bear those values in mind. They change a bit throughout the duration of the practical. M1 and M2. The M3 is going to be 40. And then M1 here, then M2 slash and M3. So just bear those details in mind. It may help you understand how to essentially calculate N. But essentially, you're multiplying by the specific values that you have from essentially the first two rows of M and C divided by R. So it's quite straightforward, and then that gives you N, your capital N, based on the other formulas, which you would see. Actually, for correction, you have N. So on page 79, to calculate N, to calculate your standard deviation and variance, you can make use of the bottom two formulas, irrespectively on, so essentially variance S2, standard deviation, lowercase s, on the bottom or end of page 79. And then the confidence intervals, which is very important, folks, that's going to be plus or minus, so N plus or minus 1.96 times the standard deviation. So your confidence interval, 95%, which refers to essentially how confident your mean is. You're 95% confident that your mean is found within that range. And that would essentially be your N plus or minus 1.5, sorry, plus or minus 1.96 times your standard deviation. So SD, it shows S, lowercase s in your manual, SD for short. And essentially that reflects your essentially confidence within a range in which your mean should be located. It may not always appear to be in this location, right? But you're 95% confident that it should be within this range. So bear that in mind. It comes out in the range because you have an error of plus 1.96 as your upper limit times your standard deviation, or 1.96 minus or divide, like essentially subtracting N by 1.96 times the standard deviation. So you want to bear those values. This is your upper limit. And then your lower limit is underneath. Both technically you should know based on the calculations, but you will be able to tabulate and essentially illustrate those values on Excel, or to graph them out and then send them to me, or essentially do them on Quarkus as your lab your final assignment for the lab. And that's shown here. And that's where you're generated essentially. Clean up. We've already done this, hopefully. How can I get an update? Are all groups or almost all groups done the exercise? Have we cleaned up our benches? Can I get actually I'll look at each group's values in due time. I look at your on the practical.
Step three. That was you. I can remember here.
I mean, just for.
But don't get changed. Oh, yeah. Yeah, no, I was just buying a lot of it. I've got a class is free. You do that first month.
Just make sure that you adjust. You have to put it all inside and you can.
Yeah.
Pick it up until they have only beans on the top. Yeah.
I've banned pardons. I've banned pardons. I don't know if you're thought.
Well, I want to make sure. Close on that end, if your confidence intervals, the upper and lower limits should be equal. Meaning that like, it will take you, like, there should be symmetrical relative. So if it's, let's say, let's say your mean is 75, and the upper limit is 100, you should expect the lower limit to be, sorry, 50, that means the 25, gap. Let's bear that in mind. Like the value should make sense for you. Charlotte, can I ask you, what do you guys study?
Oh, yeah. Oh, no.
Okay.
Yeah.
All of your student classes slide up in the next 2 minutes. Just please bear in mind that even if you see right outside, better if you're back.
No, I don't laugh.
You gotta go right. You gotta go right.
Before we do this lab, I want to make sure that all pay attention because it's not only the class, you exercise 3 as well for the full class that we do right now in class. Save yourself the time. Because it is good. So bear in mind, I hope you guys complete exercise 3 as well. If you're done, class for 4, that's what we can do in the time. And then, so my goal for you guys is that you'll have a lab review tomorrow. So try to come online. I've posted the Zoom link 2:45 to 4:45. I'm going to talk about labs 4 to 6, and hopefully, inshallah, you guys will get a step ahead, like an edge ahead, essentially, for preparing for your lab exam. It's 1 hour, but you're given a window from 2 to 4 on Friday. So inshallah, see how you can best make use of your time. I can also show the slides. Just as a reminder, 9 to 10 multiple types of questions for multiple choice, like select all of the above, or like a checkbox type of question, numerical questions based on calculations. You have matching questions. So bear in mind that you know the terms, you know the definitions, you know the calculations, so that way you could do them easily. And follow with what's shown already on Quercus, so you know what you have to do to prepare. As well, try to complete understanding these concepts, the materials that I've talked about from the lab reviews, but also from what you've seen from the past. Look through your lab manual in advance before our review, but even before you are able to actually undertake the lab exam, because you want to make sure that you're familiar with the equations, the interpretations, the observations that you would normally expect. Let's say for Daphnia, the Q10 values. Q10, by the way, indicates the level of sensitivity of a biochemical reaction on temperature. So like we looked at physiology, the physiological heart rate of Daphnia as it changed with changes in temperature. So a Q10 value that's closer to 2 indicates it's more sensitive, i.e. heart rate is more sensitive to changes in temperature. And if it's lower, closer to 1, then it's less sensitive. And so that's a relative scale for Q10. When it looks at the other parts of flowers and their morphology, remember eudicots, monocots. We talked about that in the review as well. Eudicots have sets of filaments, or like essentially the sepals or petals
that are in sets of 4 or 5, and monocots have sets of 3. Remember the primary and secondary types of tissues. You have the lignin plus cellulose that refers to a bluish green coloration. That's for secondary cell walls. And then primary cell walls are pinkish purple. And you would see this from the TBO, toluidine blue O stain. So you have those values, estimates of population size. You have a formula there, rate of growth. Pretty much what you recall from the formula that we did today on Excel. Using this formula, indeed, ln n2 minus ln n1 divided by time, right? I'll keep an eye on you guys accordingly. But make sure that you guys are able to understand this so that way you don't make mistakes, right? It's not log n2 minus log n1. It's ln n2 minus ln n1. They actually have, if you recall from math class maybe, the difference between log and ln. Anyone know the difference? Yeah.
So like log is 10,
Yeah.
log is like E.
Yes. So this is like E is another value, and that's actually 2.6, 1, 4, something, actually 2, 3. And then it just continues on. It's a natural value or number, but essentially make sure that you are using this for population growth rate. Other labs, like when I teach microbiology, they'll use like semi-log paper, and that's where you use base of 10 of log. But just bear in mind that log has its own unique value for counting and deriving certain rates of effect. And just as an example, like if you were to do this, let's say, and switch ln with log, you would actually get a different value. At least that's what you would expect. So just bear in mind that when we're dealing with the values, the equations, the exponential growth and growth rates, indeed, you want to make sure that you're following through with the right formulas. For indeed, that's the best way for you guys to understand how to perform best during the practical and for your lab exam. Many of you should have, I'm hoping, a ln. Yeah, so some scientific calculators have ln also provided. So just make sure that you're able to separate and know that, okay, I'm looking at, let's say, ln 3 minus ln 1. That gives me 1.0, like 1.1 essentially, 1.0986. Whereas if I did the same and I did log 3 minus log 1, then you actually not get you wouldn't get the same value. So just bear those values, those differences in mind. You get like 0.47. So the values differ. That's why you want to make sure that you're doing it correctly. And if you have questions, let me know. But you want to do this independently, focus on what I've talked about, attend the review seminars. If you need, like if you have an exemption, then I could send you, just let me know if you need further tips. But I really do hope you've enjoyed much of what we've done through this lab. There's still a bit of time. So let me just follow up with the last group. What was your last R3? Can I confirm? 19. Let me just confirm. I'm going to go back and look. We'll stay for another 2 minutes, so don't worry. Just like another 2-ish minutes. The slide needs to open up again. Whoa. That's me being recorded. So don't worry about you guys. If you find yourself being recorded, let me know. Give me just 1 moment. Well, let me briefly talk now. Just briefly, I want to highlight something about your graphs. So there's 2 sets of graphs. One is going to be, let me just talk to you guys about it. So yeah. Oh, you can't see it from there. Okay, I'm not even gonna look at. I'm just gonna talk to you guys. So look at page from your manual. Look at page 82 at the very bottom. Your goal is to have a detailed figure caption with the axes, the number of individuals. But essentially for this graph, you're going to have essentially the confidence intervals, 95% confidence intervals, the lowercase n, which is your absolute, likely around 75. And that would be shown as orange here. And then your capital N, which is what the formula I've shown earlier would indicate from the three parameters that you would have gotten for each of the experiments. So you're doing that for all three. That's what your table would show. And then the other graph that you have, inshallah, is going to be your two graphs actually. And I'm going to show it to you here. I can't actually show you because you guys have to do it yourself. But the idea is that you're going to plot all of these values. So imagine this was your exercise. You get all of these values, repeat the process. And this is that's why I want you guys to be a bit careful. So I have a trick for this formula. It's essentially star 4, star 2, 3. It looks like a cash sign for you guys. Oh, oh, sorry, this is supposed to be 8. 8. So, oh, interesting. Okay, so it didn't do what I was planning. But nonetheless, let's go back just so you guys understand. The idea is that you would have plotted these out, taking the means for each of them, the averages for each rep, or all 3 reps per condition or treatment. Then you would essentially do the percentage, which is, remember, the mean growth rate of your experimental treatment divided by that of the control. So make sure that you're doing it the right way, i.e. if this was 1, hypothetically, or 0. Make it reasonable, 0.012. Let's say this was your growth rate, you would do 0.12 divided by 0.283, and that is your percentage. Bear in mind as well that you put this in percentage form, and make sure that you have your tables with captions and the figures that you would make. And the figures for your lab 4 for your final assignment include both your mean growth rate across the conditions of NaCl concentrations, and then one separate line graph for your percentage growth rate relative to the control. So bear those 3 different values or 3 different graphs in mind. You need captions once again, and make it detailed with the species, with the aim, objective, and highlight. This will probably be one of my final notes for you guys for the lab. Make sure you have for each figure a title, what you're exploring with specific details of the species, the conditions, or the parameters at play, right? So it would be with the units, by the way. So NaCl percentage, right? So that's your units with percentage. Second is your methods or condition, and a brief result. So you're highlighting the conditions, a bit more of what you've done. You know, they were done after 14 days, testing or examining the changes in the number of thalli in 200 milliliters of diluted NaCl in aquatic pond water, minus the control, where it was only 200 mils of aquatic pond water, and then briefly a summary of your results. I know that may have been a bit longer than expected, but the ideas are ahead of schedule, and you actually finish, ahead of time. So I'm gonna just highlight that. That's what you need for each figure, and this is actually replicable to your future labs. When I teach microbiology, I see the exact same thing for the FLRs. And in that case, it's even a bit more strict. So you need to make sure, even for your labs here, you have like 1 mark for each figure. I am taking note if you're actually being specific for your caption. So please bear that in mind. But with that, Subhanakallahumma wa bihamdik, nashhadu an la ilaha illa anta, nastaghfiruka wa natubu ilayk. That's my closing supplication. So I hope you guys have an amazing time. Lab 4 is coming. Lab 4, I'm saying Lab 4 so many times. I think there's a reason. We were on exercise 4 from the beginning, but essentially, let's see, is there anything else? Everything's cleaned up? Everything's organized? Okay. I'll give you guys 1 more minute to think about BIOA02. Go back to the feedback quiz if you haven't, or the feedback survey if you haven't done so already. It's on Quartus as well. Think about AO2 for a moment. Think about what I talked about at the very beginning. Think about, and as I've touched upon before for the previous review, I want you guys to think about integration, implications, investigation, inventions. And from my Deen of Islam, trying to implicate that for you guys to know, okay, how can you live the lives authentically, not feeling like, oh, like I'm like in the sciences, a lot of things are secular and a lot of things are like to some degree very like hard. So I want you guys to, feel relaxed. You have 22 minutes. Think about what you've learned. This is what I've taught people from microbiology. This was one of their questions for their reflective practice. What have you learned from AO2? What have you learned? Have you learned about the sciences? Have you learned about beautiful different creations in the pavilions in Toronto Zoo? Have you learned about how heart rates change in Daphnia? What have you learned about winter wheat growing up and thalia for lemnas, growing up larger to numerous quantities? What have you guys learned thus far? Think about those questions. You can actually share them with me at any time. I want you guys to think about it though and have an amazing day. I actually have a rice bead here, so have a rice day. So have a nice day. Have an amazing day, you guys. Are there any questions? Any final reflections before you guys leave? No? Anything that you wanted to share? Any suggestions, considerations that you want to give to me? Yes.
Yeah. Particularly like everything that you learn, it's more so like what you learn is what you're most interested in. Because I think it's a good perspective to take like the labs that you found most enjoyable and most fun is because you're most interested in it. So maybe when you're looking at a future pathway, you want to be looking for something that's similar to the lab that you enjoy.
That's a solid point. And I always just want to hear one more student share another like reflection or thought for consideration. Any other students? Funerals? Believe it or not, every time you guys do something with organisms, I'm very particular on knowing how we're gonna, take care of them. Daphnias, for example, they're going down the drain. They may not actually have died, right? That's my hope. Even when it comes to the plastic lids or the cups for today, I'm not gonna put them in the garbage. I'm gonna put them outside in the recycling. So like for me, I really care for the environment. That's part of my deen as well, my religion. And so I care for every organism. So even you guys maybe didn't see this, but at the zoo, there's one squirrel that came to all of us, or like a bunch of us, and I was like asking for food. I literally took food out of my plate, my bag, and I was fasting, but I still gave it food. So like you never know. You'll have so many opportunities to interact with some amazing species and some Daphnia, as like Charlotte and her team saw, were pregnant. So you never know what you see, and you'll come to really appreciate the different organisms. So with that, I hope you have a fantastic time. Prep well for the lab exam. Let me know if you have any questions. Everyone has my email, by the way. Yeah? Hopefully you have seen my inbox messages. Take care, everyone. Have an amazing day. And I look forward to seeing you soon, maybe in the future. And if you guys have questions about like med school or need about like any tips or tricks for references or like summer research positions, just let me know. I'd be more than happy to see what I can do. There may be things I'm not aware of, but I'll try to support however best I can. But take care, folks. Let me know if there's any questions. There was one other thing I wanted to tell you guys. You may have it as a note. You can begin. Wait, no. There's one other thing. It's not that, yes. So I've adjusted the marks for you guys for labs 1 to 3 for assignments. Let me know if you got a grade that's lower than what you had from before. And I think there was one other thing, career-wise. Yeah. So let me know if you guys are like needing some help for like upcoming summer courses, or if you like want to get like a work study position, or if you want some tips on choosing a program, because now you guys are looking at cost probably on Acorn. So let me know if you guys have any questions, need any tips. More than happy to support in any way possible, shape, or form. Take care, folks. Have an amazing week, and happy Friday. You guys are gonna enjoy your Friday, hopefully. Take care. Have an amazing week.
Good game.
Good game.
Like when you finish the game at the end. I was gonna ask you.
By the way, if anyone wants to shake my hand, like I'll be
Shake it
mindful.
once. Twice. 3 times.
But don't worry, I'm as familiar as with him as I would be with any of you. So don't worry.
I was gonna ask you about first help with some of these.
If I can, so like I was thinking more like if you have
I have to speak on a second, I'll tell we email you. That'll be amazing.
I'll do it right now. Actually. So if there are folks that, let's say, want some research, I just sent one to some folks earlier, so I'll try to share it with others here as well. Actually, it's better offline. I want to make sure that I shut off some things. Okay, so let me share this with you. Yes, and you guys can take photos as well. And if you need any support, let me know once again. Okay, can I get your email?
Yeah, so. Soon in the summer, when I get this.
You know what? Actually, I have another idea. Send me an email, and then I'll see what to make it more like tailored and targeted. Because there are too many. I mean, it's not to say that there are too many opportunities, but to make sure that you guys have the right experience. That's something that I learned. Like, talk to me about like what your interests are, what your aspirations are, what program you're in.
This majority of that would be neurocentric.
Okay. You said OB?
Yeah.
What was that? Okay.
So like, you know. Thanks so much.
Everything okay? Everything okay? You guys can next time. I'll see you. Not in my time. Hey, Josh.
I just wanted to thank you for the semester. I just all kind of out of it, but that's fantastic. You're very sensitive, you're very, I think that sort of, sort of like. This is wondering for future stuff, like I called emailing about research and stuff like that. But during the summer, I was working, like I worked part time up in town, like 30 minutes away or whatever. I was just wondering if you know anything about like volunteer opportunities or research opportunities for first years. I'm also just looking to get more creative, just involved around campus.
Okay. So campus, by the way, is a better industry.
Pardon?
Do you think like working on campus, the ground, like closer to you, or like
30 minutes more. Like I'm up in Stouffville, I'm just.